So hello there, this is Anurag Pradhan. In this video, I'll be teaching you about PCR in little more details. So more about practical aspects of PCR, like how we perform PCR in real life in laboratory conditions. So let me quickly summarize what PCR is. PCR, polymerase chain reaction, and the steps involved in PCR are denaturation, annealing, and synthesis. And the conditions at these steps are: during denaturation, the temperature is about 95 degrees Celsius around, approximately. During annealing, it's about 65 degrees Celsius, and then during synthesis, it's 75 degrees Celsius. And then the duration of each steps is about 30 seconds for denaturation, annealing 30 to 60 seconds, and for synthesis, it's about it depends on the speed of DNA polymerase which we are using in PCR. So let's suppose it takes 1 minute, and let me represent everything diagrammatically. During denaturation, this double stranded DNA is converted into single stranded DNA. During annealing, the RNA primer binds to the specific site, and then during synthesis, this is the single stranded is extended further to make double stranded DNA, like this. But theoretically it seems simple, but in real life, in experimental condition, it's not that simple as it seems. Why? Because let's suppose our DNA, or the gene which we want to amplify, is really long. Okay, this is double stranded DNA and it's really long. So as you can see that during denaturation, we are allotting the time to be 30 seconds. So it's possible that in this 30 seconds, this long DNA doesn't completely get converted into single stranded DNA. So let's suppose some part of this double stranded DNA is still left to be converted to single stranded DNA. In order to overcome this problem, we go with a new step in real life that is called initial denaturation. Initial denaturation. During this step, the temperature is around 15 degrees centigrades, 15 degrees Celsius. So this is the extra step added in real life conditions. So in real life PCR in laboratory will be created with different procedures, and this is one of them. And then at last, there is a new procedure called synthesis extension. Let me check. Final extension. So let me tell you why we go with final extension. To understand that, let's consider this annealing step, and here you can see that it takes about 30 to 60 seconds. So there is a possibility that the DNA polymerase which we are using might take a lot of time to recognize a particular site, and then after that it sits on that single stranded DNA. The cycle continues, and then it goes to synthesis stage without the completion of annealing state properly. Or during synthesis state, it's possible that as annealing took time, so it's possible that during synthesis stage, uh, the second strand, the incomplete strand, is still incompleted. So it's possible like this. Let me show you this thing by diagram. So ideally what we are supposed to have is that we want this single stranded DNA to complete fully in order to form double stranded DNA, but in real life situation it's not as simple as that. Okay, so in real life it might happen that We expected this, but the result was this. We might end up with these results. And here you can see that we have incomplete ends. Incomplete ends. So in order to overcome this situation, the final extend procedure is done, and the temperature at which it is done is approximately 97 degrees Celsius. So during this step, all the DNAs that are incomplete, whose one of the strand is incomplete, is extended during this stage. Okay, so that's how we overcome the errors, the possibility of errors during the steps of annealing and synthesis. That was all about real life situation in laboratory conditions. So in there you will be greeted with this, this, and another stage called storage. As the name implies, at this stage everything is complete. So initially we go with initial denaturation, and then we repeat these cycles, and we end up with this final extension procedure in order to overcome the single strand errors and then the storage final completion. You must be wondering that why don't we add the time here during denaturation procedure in order to overcome this difficulty of incomplete denaturation. But the answer to the question is, if we add more time here, it would take really long time, because we are repeating these cycles many times, so this time will be multiplied, so it would take really long time. So it's not ideal to add extra denaturation time here. So to overcome that problem, we add this time to this initial denaturation stage. So the sequence is, we got our DNA sample in an Eppendorf tube or any tube, and then we perform initial denaturation stage, and then we just went into this cycle of denaturation, annealing and synthesis. Let's say we performed this for about 30 cycles, and then after completing these cycles, our PCR machine goes to final extension stage in order to overcome this type of problems or errors, and then storage final condition. So that's all about PCR in real life situation. Thank you very much for watching this video.